Hello, I'm Dr. Mark Darcy, and today we're going to be talking about the digestive system. So let's get started with that. Okay, so our aim today is to understand the anatomy and the physiology of the digestive system. I'll go through what digestion and absorption are, essential nutrients, parts of the digestive tract, physical and chemical mechanics of digestion, and a breakdown of carbohydrates, proteins, and lipids. So first, a few definitions. An organ system is a number of organs working together, such as the digestive system, which consists of structures such as the salivary glands, the esophagus, the stomach, small intestine, large intestine, amongst others. An organ is a collection of tissues working together. A tissue is a collection of cells that are similar to each other and perform a common function. In relation to the digestive system, ingestion is the act of taking in food via the mouth. Absorption is when small molecules, ions, and water diffuse or are transported into the circulatory system. Egestion is the process of passing undigested material out of the body. Nutrients can either be organic or inorganic. Organic nutrients include carbohydrates, proteins, lipids, nucleic acids, and vitamins. These are the building blocks and cofactors of enzymatic reactions. Inorganic nutrients are minerals such as calcium, copper, iron, etc. Minerals function as cofactors in enzymatic reactions and provide action potentials. So what is digestion? It's basically the process in which large molecules are broken down, hydrolyzed that is, into smaller molecules so that nutrients can be absorbed and waste products excreted. Many large molecules are polymers. Polymers are large complex molecules composed of long chains of monomers, small molecular units. So a macromolecule might be something like a protein, which is composed of amino acids bound by peptide bonds. The enzymes that break down protein are proteases and peptidases. They hydrolyze those bonds, which basically means adding water. Polysaccharides, such as starch and glycogen, are broken down by amylases and carbohydrases into monosaccharides, so single sugars, such as glucose. Nucleic acids are broken down by nucleases into nucleotides, nucleotide being a phosphate, a sugar, and a nitrogenous base. Fats are broken down by various fat-digesting enzymes, such as lipases. However, emulsification is often required first, which requires bile salts. They're broken down into glycerol and fatty acids. Now, there are different classes of what we call macronutrients: carbohydrates, proteins, lipids, nucleic acids. Dietary sources, well, they're listed here. Carbohydrates, all food sources, especially starchy plants. Function, energy source, components of some proteins, source of carbon. Symptoms of deficiency, muscle weakness, weight loss, lack of energy. Proteins, sources, all food sources, meat, legumes, cereals, roots, anything organic basically is going to have some proteins. Function, provide amino acids to make new proteins, build muscle, can be used as an energy source in extreme conditions, although protein is not the primary energy source in the body. That's carbohydrates and fats. Symptoms of deficiency, weight loss, muscle loss, weakened immune system, infection. Lipids, dietary sources, all food sources, fatty meats, dairy, plant oils. Function, major components of cell membranes, energy source, thermal insulation, and to produce the steroid hormones such as testosterone and estrogen. Symptoms of deficiency, hair loss, dry skin, hormone, and reproductive issues. Nucleic acids are found in all foods. All foods have got RNA and DNA. Function, provides sugars, bases, and phosphate that can be used to make DNA, RNA, and ATP. Symptoms of deficiency, none really, because the components can be synthesized by cells from amino acids and sugars. These are the major nutrients Then we have essential nutrients. Essential nutrients are nutrients which you cannot make yourself, so you must get them in your diet. There are 9 essential amino acids, for example. These are amino acids that are required for building proteins but cannot be synthesized, so they must be acquired through diet. We can convert some amino acids into other amino acids, but the essential ones we cannot. They must be in our diet, and if we are deficient, there will be symptoms of this deficiency. Essential fatty acids. Again, fatty acids such as linoleic acid and omega-3 fatty acids that cannot be synthesized by the body. These fatty acids are stored in our adipose tissue. We have a subcutaneous layer of fat, adipose tissue, under our skin, and a visceral layer under organs and tissues. The subcutaneous layer, that's less bad for you, basically. Obviously, having too much fat is unhealthy and bad for you in many ways. But the subcutaneous fat layer is not so bad compared with the visceral layer. It's a visceral layer around your organs and tissues that can cause issues, that can damage the liver, the heart, and other organs. Essential vitamins, important organic nutrients acting as coenzymes for many metabolic and biosynthetic reactions. Vitamins fall into 2 groups: the water-soluble vitamins and the fat-soluble vitamins. Water-soluble, soluble in water, so something like vitamin C, you can take in water, basically. It will dissolve in water, and you can absorb it the same way you absorb water. Fat-soluble vitamins, however, need to be absorbed with fat. Minerals, inorganic ions required for normal functioning, required as cofactors and part of some enzymes. Calcium for bone, muscle, and nervous system function. For example, you need calcium for muscles to contract and for neurons to release neurotransmitters. Sodium and potassium for generation of action potentials in neurons. Sodium and potassium are also involved in the sodium-potassium pumps that we have both in neurons and also in our kidneys and our intestine. Here is a brief overview of the physical structures in the digestive system. We have the pharynx or throat. We have the oral cavity or mouth, the uvula or tongue. We have the salivary glands, which come in pairs: parotid, submandibular, and sublingual. We have the esophagus leading to the stomach. From the stomach, we have the small and large intestine. Small intestine consisting of duodenum, jejunum, and ileum. Large intestine consisting of the transverse colon, the ascending colon, the descending colon, the cecum, the appendix. We also have the rectum and anus. Accessory organs involved in digestion would be things like the liver, the gallbladder, and the pancreas. These connect via the bile duct and the pancreatic duct into the small intestine. Now, when it comes to digestion, when you swallow food, the first thing that happens is that food will move down the esophagus. This is a tube that moves food from the mouth to the stomach using muscle contractions known as peristalsis. The muscle around the entire intestinal tract is smooth muscle. It contracts rhythmically, and this is known as peristalsis. Mucus is secreted from tissues in the walls of the esophagus to lubricate the food's passage. Circular muscles contract just behind a bolus of food, pushing the bolus towards the receiving section, where circular muscles are relaxed. This is repeated in the next section, moving food throughout the intestinal tract. Now, when the food gets to the bottom of the esophagus, we have the lower esophageal sphincter, which allows the food to enter the stomach. This can sometimes go into spasms, which can cause acid reflux. The stomach itself is a small sac, in some cases a small sac, in some cases a large sac, with lots of folds, so it can expand. It can hold up to 4 liters of food and liquid. Now, if you keep eating a little bit too much, so you're full, but you put that extra chip in, that extra cake, the stomach will actually stretch to accommodate that over time. And as part of what makes you feel full is that bloated feeling that you get in your stomach as it stretches. If you do keep stretching your stomach, it will actually take more food for you to feel full. which can become a vicious circle, as for this rotund chappy on the screen here. Stomach walls, they secrete gastric juice to break down food, consisting of hydrochloric acid. That actually sterilizes food as well as beginning the breakdown process. There's also an enzyme called pepsin, which is a protease, breaks down protein. It's secreted into the stomach in an inactive form called pepsinogen, so that it doesn't digest the stomach cells themselves. And when it gets into the acid, it's converted into pepsin, the active form. There's also mucus in the stomach. Pepsin hydrolyzes proteins into smaller polypeptide chains, only works in acidic conditions. Peristalsis of the stomach turns food into acid fluid, which is chyme. Chyme is a mixture of acid and partly digested food. The small intestine has three parts, the two main parts, the duodenum and the ileum. Chyme is moved to the small intestine by peristalsis. In the duodenum, bile, which is alkaline, and pancreatic juice, so bile created in the liver, stored in the gallbladder, released via the bile duct into the small intestine, and pancreatic juice created by the pancreas, pancreatic juice consisting of digestive enzymes and bicarbonate and hydrocarbonates to neutralize the stomach acid, is released into the small intestine and neutralizes the chyme and begins to break down food into smaller molecules. So the duodenum is basically where the pancreatic juice is secreted and where digestion begins. But really what's happening there is you're breaking down the larger macromolecules into monomers. That's happening in the duodenum. Everything mixes together, the enzymes, the chyme, the pancreatic juice, and begins the breakdown process. Then the food goes to the ileum. In the ileum, there are small soluble molecules, such as glucose and amino acids, broken down in the duodenum. They can be absorbed through the villi lining the gut wall. The villi are these big folds in the small intestine, in the ileum, that increase surface area. The cells on the villi have folded membranes known as microvilli. All of this increases surface area to facilitate diffusion. Within the villi, we have blood vessels. We also have got neurons, which help control digestion, and we've got lacteals, which then join with the lymphatic system to carry fats and a few other substances into the lymphatic system, where eventually they will be emptied into the circulation. The blood vessels that absorb nutrients, they end up joining to the hepatic portal vein, which carries them to the liver so that any toxins can be removed. The large intestine, the colon, has a folded wall for large surface area to absorb remaining salts and water, basically to dry out the poop. It also contains lots of bacteria, many different strains of bacteria, and most of them perform very useful functions. They produce vitamins like vitamin K. They help to digest fiber to extract vital, essential nutrients. The more of the good bacteria you have, the healthier your gut is and the healthier you are. Of course, if you don't feed the good bacteria with fiber, the populations will get lower, and this will lower your ability to produce those essential nutrients and to digest them, which will have systemic effects. Regular courses of antibiotics can lower the biodiversity in the guts. In the West, for example, we have very poor gut health generally, compared with in developing countries where antibiotics are taken to a lower degree. Well, in some developing countries. Actually, in India, very high amounts of antibiotic are taken during childhood, and this can have a detrimental effect on the gut. The rectum stores feces. The expansion of the rectal walls causes the stretch receptors within the walls to stimulate the urge to defecate. So in terms of regulation of digestion, neurotransmitters from neurons and hormones from the endocrine glands control the volume of saliva produced the amount of acid produced in the stomach, the timing and amount of secretions from the gallbladder and pancreas, the rate and strength of muscle contractions along the alimentary canal. The nervous system is key to this. This affects activities of the digestive system in two ways: local control of muscle and glandular activity by neurons within the alimentary canal. This is known as the enteric nervous system. Basically, the nerves in your intestinal tract are semi-independent of the rest of your nervous system. They're known as the enteric nervous system. If you add up all the neurons in your gastrointestinal tract, you basically have the equivalent of a cat's brain in neurons. Then you have long distance regulation by the brain. So local control, neurons interact with nearby smooth muscles, glands, and epithelial cells. Stimulation of neuronal activity at one point along the alimentary canal leads to impulses transmitted up and down the canal to facilitate movement of the food and digestion. When food enters the small intestine, it is stretched. Neurons in the intestinal wall are activated with impulses sent to the stomach muscles. This slows the contractions of the stomach, slows the rate of chyme moving into the intestine, which gives sufficient time for digestion and absorption in the intestine. The alimentary canal regulates its own function independent of the brain. Now, this relates actually to speed of eating. If one eats very quickly, then food will not have enough time to enter the small intestine and slow down the movement of the stomach before you have eaten too much. Whereas if you eat slowly, food will have time to enter the small intestine, which will send signals to the stomach to slow the churning and relieve hunger. So if you're trying to lose weight, try to eat slowly. Okay, one way of doing that, there's a businessman and celebrity in the UK called Lord Sugar, and he basically lost quite a lot of weight by simply taking a very small fork with him whenever he went to restaurants, his own little child's fork, which forced him to eat slowly. And therefore, he would feel full because food would have time to enter the small intestine and slow down stomach movement before he'd finished the meal. Apparently, it worked very well for him. Okay, so long distance control. The brain communicates with neurons in the stomach and intestinal walls, influencing movement and secretory activity of the alimentary canal. Stimuli such as sight, smell, and taste activate digestive functions even before food reaches the stomach. These act as feed-forward mechanisms for the brain, so the brain can prepare for digestion. Now, this means that there is a kind of issue in certain ways. So, for example, if you drink a diet drink, like this one here, then the artificial sweeteners in there, they do still taste sweet, and this will stimulate the production of things like insulin and digestive enzymes. However, because there is no sugar in there, there's nothing for them to act upon, which can basically result in lowering of blood glucose, because the insulin will lower the amount of glucose that's already in your blood, which can lead to hypoglycemia and can also lead to hunger. So no free meals, so to speak, or drink. Hormonal system. Hormones regulate the rate of digestion, secreted mainly by cells in the epithelium of the stomach and small intestine. Chemical substances in chyme stimulate cells in the stomach to release the hormone gastrin. Gastrin stimulates smooth muscle contractions and production of HCl in the stomach. Arrival of chyme in the small intestine stimulates release of the hormones cholecystokinin, CCK, and secretin from internal epithelial cells. And it works like this. The stomach secretes gastrin into the blood in response to a meal. So basically, breaking down of chyme will cause the secretion of gastrin. This will go into the bloodstream. Gastrin will then return to the stomach and stimulates muscle contraction and acid production. The partially digested food, the chyme, moves into the small intestine and stimulates the secretion of CCK and secretin into the blood. CCK and secretin stimulate the secretion of digestive enzymes and bicarbonate and hydrocarbonate ions from the pancreas into the small intestine to neutralize the stomach acid. CCK also stimulates contraction of the gallbladder, which then releases bile into the small intestine to aid with the digestion of fats. In terms of chemical digestion, this is enzyme mediated, and it involves a process called hydrolysis, breaking of chemical bonds, such as the glycosidic bonds between two glucoses in the starch. Hydrolysis breaks down large macronutrients into smaller molecules. Glands along the digestive system release enzymes to break down food. For example, sucrose into glucose and fructose. But this all starts with the salivary glands. There are three main pairs. There are the parotid, the sides of the mouth, sublingual under the tongue, and submandibular under the jaw. They secrete saliva, which contains mucus, mineral salts, salivary amylase, which breaks down starch, into maltose, which is a disaccharide consisting of two glucoses. Other functions: saliva also helps lubricate food, making swallowing easier, kills bacteria with antibacterial compounds. So we have polysaccharides, such as starch, broken down into disaccharides and monosaccharides. So for example, starch is broken down by amylase into maltose, a disaccharide, and maltose is broken down by maltase into two glucoses. These are what we call catabolic enzymes that perform hydrolysis reactions and break things down. But we have anabolic enzymes as well, which can stick things back together. For example, sticking glucoses together to make glycogen, which can be stored in our muscle and liver until it's needed. And then hydrolysis enzymes can break it down again. Sucrose is broken down by sucrase into glucose and fructose, and lactose is broken down by lactase into glucose and galactose. Although in some populations, lactase is only produced in childhood. For example, in most populations in the Far East, you stop producing lactase as an adult, and therefore become lactose intolerant. In other populations, such as Northern Europe, we tend to produce lactase our entire life. The pancreas and liver releases pancreatic juice into the duodenum through the pancreatic duct. It contains amylase, in this case pancreatic amylase, which hydrolyzes starch. Trypsin hydrolyzes protein. Chymotrypsin also hydrolyzes protein. Lipase hydrolyzes lipids. Also contains sodium hydrocarbonate to neutralize acidity of the acid from the stomach. The gallbladder secretes bile salts to reduce the surface tension of the fat droplets, basically turning large lumps of fat into small beads of fat that the lipase enzymes can then break down into glycerol and fatty acids. Breakdown of carbohydrates. When carbohydrates are first broken down, there's a high concentration of glucose in the small intestine than in the blood. So a high concentration gradient. So glucose can basically just move across the epithelial cells of the intestine into the blood by diffusion. Diffusion stops, however, when concentration in the lumen becomes lower than in the blood. Remaining glucose enters the intestinal epithelium by active transport, along with sodium ions. Basically, we use primary active transport to pump sodium into the intestine. This uses a sodium-potassium pump. And we basically pump sodium into the intestine and creates a high concentration of sodium. The sodium is then allowed back into the epithelial cells through a co-transporter. The co-transporter also has a slot for glucose. So as the sodium rushes into the cells, glucose is pulled along with it in the co-transporter against its concentration gradient, facilitating the absorption of all the glucose in the intestine, more or less. Sodium ions transported by active transport by sodium-potassium pump out to the small intestine epithelial cells into the blood. This creates a concentration gradient, a high concentration of sodium ions in the small intestine lumen than inside the cell. This causes sodium ions to diffuse from the small intestine into the lumen down a concentration gradient. This is a sodium-glucose co-transporter. Glucose is also carried in by this co-transporter. Glucose can then diffuse out of the cells into the blood, down its concentration gradient through a protein channel by facilitated diffusion. And this is basically showing you this general process. Enzymes on the luminal surface of the small intestine, epithelial cells, digest disaccharides into monosaccharides. Monosaccharides are absorbed into the cells by facilitated diffusion or by secondary active transport with sodium. At the other end, the blood capillary end, the absorbed monosaccharides leave the epithelial cells by facilitated diffusion and enter the blood. The bloodstream distributes the nutrients throughout the body, although going to the liver first to remove any toxins. Breakdown of proteins. This starts in the stomach, fully digested in the ileum. Proteases, hydrolyzed proteins absorbed via co-transport, just like glucose. So we have pepsin in the stomach, trypsin, chymotrypsin, elastase in the small intestine secreted by the pancreas. In the stomach, whole proteins are chewed in the mouth and then sent to the stomach. Once in the stomach, hydrochloric acid denatures proteins, unfolding the tertiary structure, the 3D structure, to reveal the polypeptide chain. And then pepsin can start breaking down those peptide bonds. In the small intestine, trypsin, chymotrypsin, and proteases continue enzymatic digestion, forming tripeptides, dipeptides, and amino acids. In enterocytes, tripeptides and dipeptides are further broken down into amino acids, which are absorbed into the blood. So amino acids basically pass into the blood, then to the liver, which regulates the distribution to the rest of the body. A variety of different transport proteins move the products from protein digestion to the mucosal cells. Some amino acids share the same transport channel. Dipeptides and tripeptides can enter the mucosal cell. Once there, they are broken down into single amino acids by enzymes in the lysosomes of the cells. When it comes to fats, there's both physical and chemical action that's required. Physical is the action of bile salts released from the gallbladder, produced in the liver. They're released into the duodenum and they emulsify lipids into small droplets. Chemical action of enzymes such as lipases to hydrolyze lipids into fatty acids and glycerol. They're absorbed by simple diffusion into the epithelium, but then reformed into triglycerides and moved into the lacteals in the villi of the ileum, and then enter the lymphatic system, later draining into capillaries. So basically, bile salts from the liver coat the fat droplets. Pancreatic lipase and colipase break down fats into monoglycerides and fatty acids stored in micelles, small beads of fat. Monoglycerides and fatty acids move out of the micelles and enter cells by diffusion. Cholesterol is transported into cells. Absorbed fats combine with cholesterol and proteins in the intestinal cells to form chylomicrons. This basically involves a smooth endoplasmic reticulum and Golgi apparatus. Chylomicrons are removed by the lymphatic system into the lacteals. As for vitamins, minerals, and water, water-soluble vitamins are absorbed by diffusion or active transport in the small intestine. Fat-soluble vitamins like A, D, E, and K follow the same pathway as lipids. Other minerals, calcium, magnesium, potassium, and zinc, and iodine, present in small concentrations are also absorbed by this mechanism. The majority of water absorption occurs in the small intestine, where membranes are very permeable to water, due to lots of aquaporins, water channels. Water diffuses across whenever an osmotic gradient is established by active absorption of solute, sodium chloride, hydrocarbonates, bicarbonates. And that basically is a summary of the digestive system. I hope this has been useful. And we will continue with another lecture soon. But for now, goodbye and have a great day.